Okay guys, let's begin. So today we're going to start our nephrology series, where today we're going to do the urinary systems, and on Thursday we're going to be doing the acid-base balance. So the urinary system is anatomically and physiologically, we have to classify how is the urinary system divided: anatomically, structurally, and physiologically functional. So anatomically, we have an upper urinary tract and a lower urinary tract. So that's why patients are diagnosed with an upper urinary tract infection and a lower urinary tract infection. And the upper urinary tract begins from your adrenal gland and it extends down to your ureters. And from the ureters and below, that is our lower urinary tract. There's a very important anatomical landmark that we must understand really well, and that is the hilum. The hilum is an indentation in which you will have the renal artery bring in 20% of cardiac output into the kidneys. And then you would have the renal vein taking out that filtered 20% of cardiac output. There's also sympathetic and parasympathetic nerve stimulation that enters through the hilum. And there is also lymphatic drainage. So that hilum is very important anatomical landmark. The basic unit of our urinary system, the urinary parenchyma, is the nephron. So this nephron represents our primary basic unit of our urinary system, of our kidneys. The nephron is responsible for filtering the blood. In addition to filtering the blood, the nephron is also responsible for reabsorbing important substances such as glucose, such as solutes. The nephron is also responsible for secreting poisonous, toxic substances that are in the blood, sending them back into the tubules. And then the nephron is also responsible for excreting the urine out into the bladder. And then the bladder will serve as a two-in-one organ. It will store the urine, and then with the proper stimulation in the proper time and place, it will voluntarily void and remove that urine. We will talk about that in more detail. So today's lab is very important in the sense that we're going to be looking again at this very important formula where the flow of fluid, whether it's gas fluid, we learned in the respiratory system that gas fluid is affected by the diameter of the airway. Also, liquid fluid, such as blood, is also affected by the radius or the diameter of the blood vessel. So today we're going to be looking at the effects of the blood flow to the kidneys and the relationship between the radius. But which radius? We have two radius that we're going to be looking at. We're going to be looking at the afferent arteriole radius and the efferent arteriole radius. So what relationship does the glomerular filtration rate have with the diameter of the afferent arteriole? What relationship does the glomerular filtration rate have with our efferent arteriole? Well, first let's define them. Let's first of all locate them. Where exactly is the afferent arteriole and where exactly is the efferent arteriole? So in the nephron, we first have our afferent arteriole bringing blood in for filtration. Then we have Bowman's capsule, where there will be a capillary full of multiple holes and fenestrations in addition to these holes, there is an additional basement membrane that is made up of proteoglycans, and there is a third layer known as foot processes, or the podocytes, that look like feet. And they serve two functions. The first function is a mechanical function, to literally block physically the passage of red blood cells and to block the passage of plasma proteins. But are they only working physically and mechanically? No. They are a physical barrier, mechanical barrier, but they are also a chemical barrier. They are negatively charged. So the basement membrane is made up of proteoglycans, proteins that are proteoglycans with amino acids that are negatively charged. Well, isn't that interesting? Who else is negatively charged? Plasma proteins. So now there will be a repulsion. So the blood that's coming into the Bowman capsule, full of plasma proteins, full of lipoproteins, carrying our cholesterol, they are all negatively charged. Who's going to be blocking them? The basement membrane, because it's also negatively charged and it will push them out. Not only is the basement membrane negatively charged, the foot processes are also negatively charged. So you have a two-layer mechanical layer to block the passage of these plasma albumin proteins and these negatively charged lipoproteins. That's why you should be really shocked if you see in the blood filtrate that there is plasma or red blood cell, because the red blood cell is negatively charged. It's blocked and pushed away and repelled with the basement membrane and with the podocytes. You should also be surprised if there is, in addition to red blood cells leaking, if there's any lipoproteins, or if there's any cholesterol, or if there's any plasma albumin, because they are all repelled physically and chemically with charges. Now, what happens is, in this Bowman capsule, we have a separation of two fluids. This fluid is going to be our urine that will collect ultimately in the bladder. So that's fluid 1, urine. Fluid 2 is going to be our blood. So now our blood is going to go, we have two capillaries, by the way, not just one. So we enter through the afferent, arteriole, and we exit through the efferent arteriole. Where do we exit to? We go to the heart? No. We go to another capillary just like this one, where we have, but we no longer have podocytes, and we no longer have a basement membrane. We just have fenestrations and capillaries. We have fenestrated capillaries, and this network will ultimately lead back into the heart. This capillary here is known as what? What is the name of this capillary? This capillary is our glomerular capillary. This is the glomerular capillary. Then the blood, after urine has been formed, the rest of the blood that is going to the heart does not immediately go to the heart. This is a portal system. We said that in portal systems, like in the hypophyseal portal system or in the hepatic portal system, you have blood going through two adjacent or two connected capillaries before it goes to the heart. What's the name of this other capillary? Maybe if I tell you what it does, you're going to remember it. This capillary filters out all of the positively charged, so that makes sense, solutes like calcium and potassium easily flow out because they are small, they evade the physical mechanical barrier, and because everything is negatively charged in the barriers, it's gonna attract them and get them to go out. But that's not the only thing. That's filtration. So we don't only rely on one process, on filtration. There is also reabsorption. So where does the reabsorption go? It goes into this second capillary. So there is reabsorption, like reabsorbing glucose and reabsorbing very important solutes and electrolytes. And not only that, there is secretion. So any more solutes that we don't need. That's how your renin-angiotensin-aldosterone system happens. If blood pressure is high, we secrete sodium so water can follow behind the sodium and decrease the blood volume. And then lastly, you have water reabsorption, and then you have excretion into the bladder. So I have lost things here, and I have lost again here, so I lose twice. I filter, I lose, and I secrete, I lose, but I gain when I reabsorb, and I gain water. And then they come back into this capillary, and then we go right back into the heart. What is the name of this very important second capillary? The proximal convoluted tubule is inside that capillary, but this is not, the proximal convoluted tubule is not a capillary. It's a tubule that is responsible for concentrating this urine. So the proximal convoluted tubule is engaging in the process of reabsorption, but eventually it feeds into this capillary, but it is not the name of the capillary. Yes? Excellent. That is the peritubular capillary. So the peritubular capillary is the second capillary that will receive the conclusion of this nephron activity. We have filtered blood, we have reabsorbed blood, we have secreted substance, and we have reabsorbed water, we've excreted. That is the blood that is going to. So if you take a sample from blood in the peritubular capillaries and a sample in the right heart blood, they will be identical. But if you take a sample from the blood of your glomerular capillary, compare it to the heart, it will not be the same, because it is just about now to enter into these 4 different physiological processes: reabsorption, secretion, and water reabsorption, and filtration. This is very important. This concept is very important. It's also important to understand that these different processes, the filtration, the reabsorption, the secretion, and the excretion, they're not the same for all. They're not the same for all substance. So, for example, creatinine, you would have filtration equal excretion. We don't want anything of it back. We want all of it out. So you would see that the number of creatinine that has been filtered out from the glomerular capillary will equal the number of creatinine in excretion. So for creatinine and only creatinine, you will have filtration equal excretion. Now for glucose, you will have filtration, but you will have reabsorption more than filtration. You will have reabsorption more than filtration. You will have reabsorption more than excretion, because we reabsorb more glucose than we excrete. So when you compare creatinine and you compare it to glucose, you will see that excretion of glucose is equal to the filtration of glucose. But for creatinine, you will see that the excretion is equal to the filtration. But that's not the case with glucose. In glucose, you would have reabsorption more than excretion, because we reabsorb more. And now this is for solutes. It is for solutes, you would have per basis. So for example, it depends on the patient. If the patient has a high blood pressure, then you will see that the sodium secretion will be more than the reabsorption. So this is just for you to understand that these processes, they're not the same for every single substance and every single molecule. If you have metabolites like medications, if you take meds, all of the byproducts of the medications will be secreted, just like creatinine. We're not going to reabsorb any of them back into our peritubular capillaries. Is that clear? This concept is very important, and how in our nephron we are able to perform filtration and then reabsorption with peritubular capillaries, then secretion, then water reabsorption, then excretion into the bladder. And also important to remember that for every single substance, we have differences into, like I said, creatinine will have equal excretion to filtration, and glucose will have more reabsorption than excretion. And is that point clear? Okay. Now, what are the factors? What are the factors that influence for me the glomerular filtration rate? So now I'm not looking at the peritubular capillaries. I'm looking at my glomerular capillary. The glomerular capillary has foot processes. It has foot processes. And then it also has a proteoglycan layer known as the basement membrane. And then it also has holes known as fenestrations. And then blood goes out this way. Okay, now, very interesting. We know that the endothelium itself is negatively charged. And you would have proteoglycans on the basement membrane that is also negatively charged. And you would have on the podocytes and the foot processes, they are also negatively charged. So blood is entering. I need to now find out what are the factors that affect for me the filtration process. Well, let's look at what pressures am I dealing with. There are two compartments here. What are they? What are the two compartments that are within my Bowman capsule? This is my Bowman's capsule, and it is made up of a capillary and what are the two compartments that are against each other? Because remember, I'm trying to make two fluids. I'm trying to make fluid that is blood fluid going into the peritubular capillaries to be sent back to the heart, and I'm trying to make urine. So obviously I have two forces, I have two factions, I have two enemies. The first one is the urine fluid, that's why I call it fluid 1, and fluid 2 is blood. So because I have two fluids, that means I definitely have two compartments. What are the two compartments? The first compartment is the capillary compartment. The glomerular capillary compartment. And it has its own pressure. Glomerular capillary pressure. Hydrostatic pressure. The pressure of the blood flowing into the glomerular capillary. It also has osmotic pressure. We have glomerular capillary osmotic, also known as oncotic pressure, and we refer to that as pi, this as P. So we have two P, we have one P that is capillary pressure, and we have one pi that is osmotic pressure. That's for the capillary, the glomerular capillary. What is the other compartment? The other compartment is the filtrate. The filtrate that is leaking out. So I have a filtrate of blood that is going back to the heart. The second compartment is going to be the filtrate that I'm filtering out to make urine. So the second compartment is what is leaking out successfully, what made it through the endothelium, chemical barrier, mechanical barrier, what made it through the basement membrane, and what made it through the podocytes and is now leaking and filling out this interspace. This is known as the filtrate. So now I have four forces. I have the hydrostatic pressure, I have the hydrostatic pressure of the glomerular capillary, and I also have the oncotic pressure of the solutes here. So I have pressure pushing the filtrate out. So the pressure of the glomerulus is pushing the fluid out. So let's designate the urine direction as the positive. So I'm trying to go into the bladder, that's my positive direction. That's my bladder. So if I'm trying to get the urine out, so the pressure of the glomerulus, is it positive direction or is it going into the blood, the negative direction? Is the pressure of my glomerulus, the pressure of my glomerular capillary, pushing all of the solutes out into the filtrate, into the urine? That we said as our positive direction. That's our positive direction. So the pressure of my glomeruli, the pressure of my glomerular capillary, that is in which direction? That's the positive direction, because now you have pressure forcing all the solutes to exit, pushing all the solutes to leave, to make their way out into the urine. So that makes it the positive direction. What about the oncotic pressure of the glomeruli? Is it also pushing out in this direction? No, it's telling all the substance, these are solutes. So oncotic pressure is exerted by solutes. So now it's telling everybody that is being pushed out by the glomerular pressure to return. So it's telling it, come back. Oncotic pressure is fighting hydrostatic pressure. So it's in the negative direction. So that is as far as glomerular oncotic pressure and glomerular hydrostatic pressure. They are working in the opposite direction. Hydrostatic pressure pushing out into the urine, oncotic pressure saying, please come back, return. Now, what is the other compartment? The other compartment is the hydrostatic pressure of the filtrate. That is accumulating here. And the hydrostatic pressure of the filtrate is obviously pushing back. It's pushing back, pushing the fluid back into the glomerular capillary. So that is in the negative direction, because it's pushing it back against the urine. And then we also have oncotic pressure of the filtrate, of the solutes that have successfully leaked out and are now accumulating in this interspace. And what they're doing is they are absorbing, they are making oncotic pressure that is absorbing all of the substances into the filtrate. So that is in the positive direction. It's very important for you to understand that we have two compartments, the filtrate compartment and the glomerular compartment. And each compartment, within it, it has two different forces. It has the hydrostatic force. Remember when we talk in a circulatory system about how when blood enters into the capillary, there is hydrostatic pressure, and that is delivering the blood into the capillary, and then there is the lymphatic drainage that is returning all of the solutes, but there is also oncotic pressure that is returning all of the solutes back into the capillary. So you must recognize that we have two different compartments, the glomerular compartment and the filtrate compartment, and each compartment has two pressures, one hydrostatic pressure and the other one oncotic pressure, and this one has a hydrostatic pressure and it has oncotic pressure, and the filtrate will be the net sum of all of these factors working together. Is that clear? Okay. This is very important. So all of these factors are working together to generate for me step 1, which is filtration. That's how filtration happens. So now I have filtered my blood. So now I'm moving to phase 2, which is reabsorption. In reabsorption, this is so important. In reabsorption, you must also remember, so now we have left the glomeruli. I'm having a difficult time saying glomeruli. So now we have left the glomeruli. And now we are dealing with, this is the tubule. This is the tubule. And this is the blood vessel. And this yellow is urine. This is the urine side. This is all the urine going into the bladder. What is this side called? This side is called the apical side. So this is the tubule. This side, that's urine. This urine, that's the apical side, also known as the luminal side, because it is the lumen of my tubule. These are the epithelial cells that, and on this side you have another epithelial cell side, like this. So you have these cells where you have paracellular, transcellular passage. So we said that if we want to reabsorb sodium, then we reabsorb through either transcellularly. So if you pass through the cell with channels, that makes it transcellular passage or transcytosis. But if you go through the gap junctions, then we said that is known as parasitosis or paracellular passage. So this is the apical side, the luminal side. Here on this side, this is the lumen of the tubule. This is the lumen of the tubule. Okay, let me make a better illustration. This is very important. Okay. Where are we now? We have filtered successfully. We have dealt with filtration. We're done with this stage. Now we are here. So, this is the lumen of my tubule. So that means everything here is urine, is going out in the urine. I'm right here now. This is urine, and this is the lumen. That's why they call it the luminal side. This is the lumen, also known as the apical side. And I have cells, epithelial cells, on both sides of my lumen. These cells have gap junctions. And I have blood vessels here. These are my peritubular capillaries returning the blood. So this is the lumen, and this side here is my peritubular capillaries. So that's the lumen, and this side is known as the basolateral side. Basolateral side. So what happens is, I have finished filtration. Now I want to deal with reabsorption. This side that is facing the core of my tubule is known as the luminal side or the apical side. And the side that's here, that is facing my blood vessels, that is known as the basolateral side. On the basolateral, this is so important. On the basolateral side, you have sodium potassium ATPase that would hydrolyze ATP for me to throw sodium to throw the sodium against concentration gradient. So I know in my blood vessels, extracellularly, sodium is supposed to be more. So extracellularly, sodium is more. Extracellularly, sodium is more. But that's why I have on the basolateral side this sodium, potassium, sodium, potassium, sodium, potassium, ATPase, that is hydrolyzing ATP to throw for me sodium from the core of my epithelial cells into where it's even more sodium. So it is hydrolyzing ATP to take sodium from where there is little sodium to where there is a lot of sodium, the blood vessels. Here, it takes sodium from where there is a little bit of sodium up to where there is a lot of sodium into the blood vessels. So that is very interesting, because now when I deplete the sodium inside of my epithelial cells, when I deplete the sodium, what did I do now? I have opened the path for secondary transport. So now sodium will start to flow down electrochemical gradient, because the sodium potassium ATPase keeps on taking sodium from here, dumping it into the blood vessel. So it makes a little bit of sodium. And since there is a lot of sodium in the urine, what happens is, for reabsorption, we're going to couple the passage of sodium flow from an area of a higher concentration to an area of a lower concentration, known as secondary active transport, to transport glucose, to transport amino acids, to transport amino acids, to reabsorb amino acids. So, do you now see? This is very important. I have used the sodium potassium ATPase to deplete the core of my tubule epithelial cells from sodium. And when sodium has been depleted here, thanks to active transport, primary active transport with the sodium potassium ATPase, then the sodium that's here in the urine, is going to flow smoothly. It doesn't know that it was because of this sodium potassium ATPase, all of the sodium has been moved into the blood vessels. It just sees there is a little bit of sodium here, and sodium moves from an area of a higher sodium concentration, the urine, the luminal side, into an area of a low concentration gradient. And who hitches a ride, who takes on a ride, is glucose. Who takes on the ride? It's the amino acids. Who takes on the ride? Chlorine. So there is a channel here known as sodium to chlorine and potassium channel that transfers one sodium, two chlorines with it, and one potassium. And that is how antidiuretics. So patients who we don't want them to have diuresis, we will block this channel. But what does the channel do? It uses this gradient like a waterfall. You know, in the waterfall, the water's flowing down, so they put a rotor. They put a rotor here, and when the rotor goes down, it makes when the water goes down, it makes the rotor spin, and there's a magnet inside there, and that causes energy. This is so important for you to understand. It's so important for you to understand that the sodium potassium ATPase is using ATP to send sodium from the core of my epithelial cells, of my tubules, into the blood vessels, and that will cause a really negative voltage down here. And this negative voltage is so nice for the core of my lumen, for sodium to flow from an area of a high sodium concentration to an area of a low sodium concentration, from an area of a high sodium chemical gradient to an area of a low sodium chemical gradient, from an area of a high sodium voltage to an area of a low sodium voltage. And with the passage of sodium, we then perform secondary active transport to reabsorb glucose, reabsorb amino acids, reabsorb solutes. Is this point clear? This is the core of the renal system, that you must understand this step right here. That's how we perform our second step, the reabsorption, glucose reabsorption. Now, secretion. How does secretion happen? Secretion happens the same way as reabsorption. The only difference is we use an antiporter. We use a counter transporter. Here we use a uniporter. So in the same direction, sodium is going into the core, it's taking glucose with it. Sodium is going into the core, it's taking amino acid with it. Sodium is going into the core, it's taking chlorine, and it's taking also potassium, and it's taking also calcium with it. The only difference is in secretion, sodium will always be going in from an area of a higher concentration to an area of a lower concentration. But in secretion, the arrow will now point the other way around, where hydrogen proton will be secreted out. But we still use sodium. We still use sodium flow inwardly so that we can secrete hydrogen out. This is so important for you to understand. That step, that antiporter, that counter transporter, that is how secretion happens. So in all cases, I'm using sodium flow inwardly thanks to sodium potassium ATPase. But for reabsorption, everybody's flowing inwardly. But for secretion, sodium is still flowing inwardly, but hydrogen proton is flowing outwardly. And that what concentrates our urine, makes it more acidic. Is that point clear? Anybody has a question about this point? Excellent. Okay. And then in the end, we have water reabsorption. So water moves. So where does bulk flow happen? Remember what I mentioned? Bulk flow, where the solutes follow water. Where does it happen? Where does this step happen? It happens right here. It does not happen in here. It happens here between the epithelial cells and your nice endothelial cells. So the endothelial cells have gap junctions too. So water will be moving transcellularly, and it can also leak through the gap junctions paracellularly. And when water leaks, some solutes will follow water paracellularly, and that is known as bulk flow. It's also known as bulk flow and ultrafiltration. It's called ultrafiltration or bulk flow. So all the water will be moving back from the lumen into the blood vessels. Okay, last thing is the bladder. We have concentrated the urine, we have reabsorbed the water by ultrafiltration or bulk flow. Then we store our urine into the bladder, and the bladder has stretch receptors that feed into spinal cord. The spinal cord is the computer that integrates that there's a the bladder has been distended, it's really full, and then it will send the output signal for the voiding and the removal of this urine. Remember that your cerebral cortex can override. That's how you're able to hold on urine. You do not urinate on yourself, and you do not cause urinary incontinence. Why is that? Because that shows you it is not only your spinal cord who is responsible of you voiding the urine. It is your cerebral cortex understanding the proper time and place. That's why babies whose cerebral cortex has not fully matured and developed yet anatomically, structurally, and functionally, what happens is they don't have the ability. They do not have the ability to control the urine, and they have urinary incontinence. Also, in the other spectrum, babies and the real elderly, due to dementia, due to neurological deficits, they are no longer able to control the voluntary control of urine void. But we will talk about that more on Thursday. For now, I needed you to understand this really well, that Sodium potassium ATPase creates a gradient for me, and that gradient for me is primary active transport that I later utilize as secondary active transport and utilize the passage of sodium from an area of a higher concentration gradient to an area of a lower concentration gradient to reabsorb and even to secrete. Okay, so now, last thing is, what is the lab is all about today? So the lab is all about what are the effects, what determines the filtration rate. So we talked about multiple factors, like the oncotic pressure, and also the oncotic pressure of the glomerulus, and the hydrostatic pressure of the glomerulus, and the oncotic pressure of the solutes in my filtrate, and my hydrostatic pressure of my filtrate. But the lab is not going to be testing that. The lab is looking at another dimension, which is what is happening in the blood vessels that bring in. So I know that this is my afferent arteriole, and I know this is my Bowman capsule, and this is my efferent arteriole that will later on carry everything into the peritubular capillaries. And this is my urine forming. So the lab is going to be today about this. This is my afferent arteriole. And this is my efferent arteriole. The lab is going to be about this formula, where the flow of fluids, whether gas fluids or liquid fluids, whether air flowing into my airways or blood flowing into my blood vessels, either way, the flow of the fluid will be directly proportional to the diameter of these vessels that are mediating, these conduits that are mediating its passage. So what they want you to do in the lab is to look at what will happen. What will happen if I decrease the radius of my afferent arteriole? What will happen to my glomerular filtration rate? Yes. Yeah. It should it will decrease. But that's what the lab is about today. If I decrease my afferent arteriole and I keep my efferent arteriole constant, how will that affect my glomerular filtration rate? Then they're gonna have you do a couple experiments. Increase it, increase. So you're gonna increase the afferent arteriole from 0.35 millimeters up to 0.50, and then you're gonna click on, remember, record data. When you record the data, then it's gonna measure for you the glomerular filtration rate, and then you're gonna look and see, oh, when it was 0.35, the GFR was this much. When it was 0.40, the GFR went up. When it was 0.50, the GFR went even more up. So now you have learned, really, there is a direct relationship. So there will be a direct relationship between glomerular filtration rate and the afferent arteriole. But when we're looking at the efferent arteriole, you will see that there is an inverse relationship, where the glomerular filtration rate will have an inverse relationship with the efferent arteriole. There is no symbol for inverse, but we use the reciprocal of the thing. So if I want to say there is an inverse relationship between the GFR and the efferent arteriole, I just put 1 on the top of efferent arteriole and I keep on my symbol. Are we clear? This is exactly what the lab is about today. How are we going to modify the radiuses, and how modifying these radiuses going to affect my GFR. They also have something really nice too, which is the pressure. So the glomerular filtration rate is also directly representing the glomerular filtration pressure. And it also represents for me my urine output. So the more I filtrate, The higher the glomerular filtration rate means the higher the glomerular filtration pressure means the more urine. So if I increase the afferent arteriole, I will increase the glomerular filtration rate. I will increase the glomerular filtration pressure. I will increase urine in my bladder. The opposite for the efferent. If I drop my efferent arteriole radius, that means I will drop my glomerular filtration rate. That means I will drop my glomerular filtration pressure. That means I will drop the amount of urine that will accumulate in my bladder. So that is what we're going to be doing today. Any questions? Any concerns? Any suggestions? Speak now or forever hold your silence. Okay, everybody click on
recording.
Yeah. That's amazing. So now, if you missed any segment, you can go back and watch it. Okay. So, let's go to, it logged me out. No, it did not. Okay, let's now go to exercise 9. Exercise 9 is the renal system physiology. Let's click now on activity 1. Activity 1 is the effect of arteriole radius on glomerular filtration. So what is my independent variable and what is my dependent variable? If I am measuring the effect of arteriole radius on glomerular filtration pressure, and I said we're not just, that's the name of the experiment, but we're not only going to be looking at glomerular filtration rate. We're also going to be looking at glomerular filtration pressure and the urine that is going to be formed. So which one is my independent variable? Which one is my dependent variable? Yes?
The radius is independent.
Excellent. So the radius is independent. So I'm always going to be plotting my radius values on the x-axis or on the y-axis? On the x-axis. So now my radius dropping. I cannot change that. But what's going to be affected by the changes of my radius, what's going to be affected by my independent variable is going to be my glomerular filtration pressure, my glomerular filtration rate, and my glomerular filtration and my urine production. Okay. The effect of arteriole radius on glomerular filtration. Number one is to understand the terms nephron, glomerulus. We've talked about those. Glomerular capillaries, the renal tubule, where I had the lumen, and the filtrate. That's my second compartment. So the first compartment is the glomerular capillary. The second compartment is my filtrate. And then there's Bowman capsule. And then there's the renal corpuscle. And there's the afferent arteriole, the efferent arteriole, and the glomerular capillary pressure, and the glomerular filtration rate. We're going to be looking at all of those. Then we must understand how changes in the afferent arteriole radius impact glomerular capillary pressure and filtration and urine production. Even though they didn't put that in there, we're going to be looking at that also. Then we're going to be looking at our second independent variable, which is the efferent arteriole radius. How changing and modulating and modifying that, how is it going to change? How is it going to change the glomerular capillary pressure and filtration? Yes?
My PCOS isn't working. It's saying that like the supply user is associated with more than one account.
Okay,
I
so
don't know. Can I work with Eric?
okay, so just speak to me in the end.
Okay.
Yeah, you can work with Eric to see how it is, and then I'll tell you how to do the experiment and send it tonight, okay? Okay, each of the million, that is a question of the pre-lab. So each of the million nephrons in each kidney contains two major parts. The first one is a tubular component. That tubular component is how we're going to form the urine, and that's how we're going to be reabsorbing and secreting. Then the renal tubule and a vascular component, which is the renal corpuscle. So there are two parts. The tubules that form for me the urine by reabsorption and secretion. And then we also have a vascular compartment made up of capillaries. So there's the renal corpuscle. So the glomerulus is tangled capillary knot that filters fluid from the blood into the lumen of the renal tubule. So that's my second compartment, the filtrate compartment. The function of the renal tubule is to process the filtered fluid, also known as the filtrate. So the filtrate is my second compartment. The beginning of the renal tubule is an enlarged end known as the Bowman's capsule. So that is the beginning of the renal tubule, or the glomerular capsule, which surrounds the glomerulus and serves to funnel the filtrate into the rest of the renal tubule. So collectively, the glomerulus and the Bowman capsule are known as the renal corpuscle. That's very important. The renal corpuscle is made up of what? Of two compartments. It is made up of a vascular compartment known as the glomerulus, and it's also made up of a tubular compartment known as the Bowman capsule. Clear? We agree? Do we all now know the significance of the renal corpuscle? That it is made up of both, not just one, a tubular component and a vascular component. Two arterioles are associated with each glomerulus. There is the afferent arteriole, feeds the glomerular capillary bed, and an efferent arteriole that drains it and eventually connects with the renal vein. These arterioles are responsible for blood flow through the glomerulus. The diameter of the efferent arteriole is smaller than the diameter of the afferent arteriole, restricting blood flow out of the glomerulus. Well, that's interesting. Intuitively, it makes a lot of sense that when I decrease the radius of the efferent arterioles, what am I doing? I'm delaying the exit of the fluid that's in my Bowman capsule. I'm delaying the exit of the fluid that's within my Bowman capsule. So it's going to lag behind, leading to more filtration. So that's intuitive. I constrict this, less is getting out, more is staying behind, that means more filtration. I dilate my efferent arteriole, everybody's gonna run out, I'm gonna decrease my filtration rate. So one of the questions is about dehydration. If you are in the desert, it's so hot, and you have hypovolemia, and you're sweating, what combination of afferent arteriole modification with efferent arteriole modification is really going to be physiological and helping you a lot? What should happen? If you are dehydrated, what should you do? Yes?
The afferent should constrict and the afferent should dilate.
Excellent. So I constrict the afferent arteriole so that I don't filtrate water, because I need to conserve my water, and I don't make a lot of urine, and I dilate my efferent just in case that this little bit amount that has leaked in, I ensure that it really leaks out as fast as possible and it does not undergo my filtration. It does not undergo any filtration. What does what is the renal corpuscle made up of? It is made up of the Bowman capsule, which is a tubular component, and the glomerulus, which is a capillary or vascular component. Okay. Consequently, the pressure in the glomerular capillaries forces fluid through the endothelium of the capillaries into the lumen of the surrounding Bowman capsule. In essence, everything in the blood except for the blood cells, red and white, and the plasma proteins is filtered through the glomerular wall. From the Bowman's capsule, the filtrate moves into the rest of the renal tubule for processing. The job of the tubule is to reabsorb all the beneficial substances from its lumen and allow the waste to travel down the tubule for elimination from the body. During glomerular filtration, blood enters the glomerulus from the afferent arteriole, and protein-free plasma flows from the blood across the walls of the glomerular capillaries and into the Bowman's capsule. The glomerular filtration rate, GFR, is an index of kidney function. So in humans, the filtration rate ranges from 80 to 140 milliliters a minute, so that in 24 hours, as much as 180 liters of filtrate is produced by the glomeruli. The filtrate form is devoid of cellular debris. It is essentially protein-free and contains a concentration of salts and organic molecules similar to that in the blood. So the glomerular filtration rate can be altered by changing arterial resistance or arterial hydrostatic pressure. In this activity, you will explore the effect of arterial radius on glomerular capillary pressure and filtration in a single nephron. You can apply the concept you learned by studying single nephron to understand the function of the kidney as a whole. These are the equipment that we're going to be using, and we'll refer to them in the lab. So, question number 1 in pre-lab. How many nephrons are in a healthy human kidney? I've mentioned that already. Yes?
10 to the 6.
10 to the power of 6, which is how much?
Million.
A million, yeah. Very good. Excellent. Which of the following list the anatomical structure in the correct order as they are encountered by the blood itself during the process of filtration? So we have afferent arteriole going into the glomerular capillary, that is within the Bowman capsule, and that is my renal corpuscle, and then we have the exit of the efferent arteriole. Are we clear that this is the right answer? Bowman's capsule is connected to the beginning of the, very good, proximal convoluted tubule. The functional unit of the kidney is the nephron. During the process of renal reabsorption, fluid and solutes move from, in reabsorption, we move from the apical or lumen into the basolateral side, which is what? Is that clear? We move on from here, from the tubule into the peritubular capillaries. Okay, so what's going to happen here? That's our blood. So now we're going to do our dialysis. This is the blood that must be filtered. And this is my afferent arteriole. Blood is going to go into the afferent arteriole, then into my renal corpuscle, which is made up of my Bowman capsule, the tubular component, and it's going to go into the glomerular capillaries, which are my vascular component, and then we're going to have our filtration happening here, and in the filtration you have all of the urine that's going to collect into my blood. This is my bladder right here. That's the bladder, this container that's in the end. And then you have the efferent radius, where everything is going to get out of my corpuscle, and then this is my peritubular capillaries. And here I will have my reabsorption, and I will have the secretion, and then the blood is now going to enter into, it's going to return into the right heart, and this represents my filtered blood that has successfully passed through the kidney. So note that the default values for the efferent radius, and the pressure, change these values to be 0.5 for the efferent radius. So let's change efferent to make it 0.5. You see that it's dilating. So let's change the efferent radius into a 0.5, and then the 0.45 is the efferent radius, 0.45, and the pressure is 90. Okay? Then click start. So now blood is entering into my Bowman capsule, and this is the proximal convoluted tubule, and this is the distal convoluted tubule. There's the loop of Henle, and then these are all my tubules. All the urine is starting to collect, and then the blood is returning into my peritubular capillaries, and that's my renal vein. And then I have restored all the blood that has been filtered. So now let's record the data. That's interesting. So now let's read the data that we recorded. Click on record data. The efferent radius is 0.5 millimeters. The efferent radius was 0.45. The glomerular pressure was 55 millimeters of mercury, and the GFR was 124, and then the urine that I generated was 200. So now let's refill and prepare for the next phase of the experiment. We have refilled. Now we have a question. Prediction. What will happen to the glomerular capillary pressure and filtration rate if you decrease the radius? If you decrease the radius of the afferent arteriole. If I decrease the radius of my afferent arteriole, everything is going to drop. So both pressure and filtration will decrease. Next, note the glomerular capillary pressure. Okay, we have done that. Okay, so now we're going to be running successive experiments where we're going to drop the arteriole every time by 0.05. So now let's go to the afferent radius and drop it by 0.05. So now we're going to have 0.45. So all of the afferent radius should read 0.45. And now let's click on start.
So the ether is still the same?
Yeah, it's still the same because we are measuring the effects of the afferent arteriole radius constriction, while the efferent is going to be constant because we don't want interference. We're testing one at a time. If you test both at a time, you're not going to be able to tell which one affected. So then in the end, we're going to come back to the efferent, but we're going to keep the afferent constant. Very good. Okay, now let's click on record data and let's compare. So when the afferent radius was 0.5, the glomerular pressure was 55, but when it was brought down to 0.45, the pressure went down to 51, and the rate went down to 81, and even the urine production went down to 179. So our hypothesis was correct. Let's now refill. We're going to be doing this until we hit 0.35. So now let's go down one more. Press on the minus signal and let's click on the afferent radius. Is now going to be 0.4. Click on start. So click on record data and let's compare. When it's 0.40, what happened to glomerular pressure? It went down to 48 compared to before. So now do we see a trend? A trend of everything going down, glomerular filtration pressure, glomerular filtration rate, very good. Okay, now let's go down to 35, which is the last one. Click on refill. You've refilled everything. Now we go down to 0.35. Now click on start. Record data. And now we're done. So we have dropped the afferent radius down to 0.35 and we see that there's an overall trend of a drop in the glomerular pressure and a drop in the glomerular filtration rate and a drop in urine volume as we have dropped the afferent radius from 0.50 down to 0.35. Okay, next. A question. What will happen to the glomerular capillary pressure and filtration rate if you increase the radius? So both pressure and filtration rate will increase. So now we're going to be doing this up to 55. So now let's increase it. So press refill first. And let's increase everything up to 0.55. Let's increase the afferent arteriole up to 0.55. Click on start.
Only the top will freeze, so.
Yeah. So now we're just increasing. So before we measured decreasing the afferent arteriole, dropping it, decreasing it. Now we're gonna look at the other side, which is increasing it. But we're always going to be keeping the efferent radius. We're going to be keeping it as constant. Record data, 0.55. Very good. Now click on refill to restore everything. So look at the data. When I increased the afferent arteriole to 0.55, the urine was increased to 212 and the GFR increased to 172 and the glomerular pressure increased to 58. Next. Now increase the radius to 60.6 and let's run the experiment again. Click on record data. So now compare a radius of 0.6 to a radius of 0.5, and you see that the urine volume has increased, the GFR has increased, and the glomerular pressure has also increased. Next, we record data, then click on refill. So now, caffeine from coffee and Coca-Cola. Caffeine consumption leads to increased urine formation. From the results in this experiment thus far, you might propose that if caffeine is going to increase urine formation, that means we have also increased, that means the caffeine dilate the afferent arteriole. Okay? Check answer. Submit question. What will happen to the glomerular capillary pressure and filtration rate if you decrease the radius of the efferent arteriole? So now I'm decreasing the efferent arteriole. I decrease the efferent arteriole, I'm gonna sequester and tighten things up, so I'm gonna have a lot of fluid lagging behind, waiting to get out, and the more it lags out, the more it's gonna filter, so that's gonna increase my filtration rate, and that's gonna increase the pressure. Both pressure and filtration rate will increase. Decrease the radius of the afferent arteriole to 0.5. So now let's go up to afferent, make it 0.5. And let's click on start to now have a baseline control values. So now we're going to start with 0.5. Record data. Okay, so we have recorded our baseline. So now let's move on to the next step, which is note the glomerular capillary pressure and the glomerular filtration rate displays and select record data. We did that. Let's refill. So replenish the source beaker and prepare the nephron for the next run. Okay. You will now observe the effect of incremental decreases in the radius of the efferent. So now we're keeping the afferent arteriole constant, and we are now changing the efferent. So let's decrease the efferent arteriole by 0.05 every time up until 0.30. So now it's at 0.45. Let's make it 0.40, and let's start the experiment and get the data. Click on record data. Click on refill. Now let's look at the values. When I drop the efferent arteriole from 0.45 to 0.40, what happened to the pressure? It went up. What happened to the GFR? Went up even more. What happened to the urine volume? Went up even more. Let's now, we're gonna drop it down by 0.05 up until we reach 0.30. So let's do one more, 0.35. Click on start. Then record data. Then let's do the last one, 0.35. Click on refill. 0.30. And then let's click on start. Okay, we did 0.30. So now let's compare. Let's compare dropping the efferent radius by 0.05 all the way from 0.45, and we will see there is an upward increase, there's a trend, an increase in everything, glomerular pressure and glomerular filtration rate and the urine volume has increased. When you are in the desert and dehydrating, which of the following arterial combinations would benefit you the most? What do you think the answer is? What will benefit you the most? Third one. Third one is afferent constriction and efferent dilation. Exactly. Submit question. And submit experiment. Submit. Okay. Post-lab quiz. If you increase the afferent arteriole radius and keep all the variables constant, the glomerular filtration rate would increase. If you decrease the efferent arteriole radius and keep all the variables constant, the urine volume, the urine flowing into the urinary bladder would do what? What would happen to the urine? Would decrease. If you increase the efferent arteriole radius, if you increase the efferent arteriole radius and keep all the other variables constant, the GFR would Who's going to help me with this one? If you increase the efferent arteriole radius and keep all the other variables constant, the GFR would, yes?
Decrease.
Would decrease. If you decrease the afferent arteriole radius and keep all the other variables constant, the volume of urine flowing into the urinary bladder would, if you decrease the afferent radius, it would what? It would decrease.
Wait, can you do number 2 again?
Okay, number 2, this one?
Yeah.
Okay, if you decrease the efferent arteriole radius and keep all the other variables constant, the volume of urine flowing into the urinary bladder would increase. Yeah, it would increase. If you constrict this guy, then you would lag behind. There will be a lot of pressure here trying to get out. That would increase the glomerular filtration rate, that would increase the glomerular filtration pressure, and that would also increase the urine volume formation. Wow. Okay, 4. We did 4. Are we done? Submit. Continue. Okay, what are 2 primary functions of the kidney? So the kidney is a very important hormonal regulator. So what does the kidney do? The kidneys would sense that there is a drop in. So the first function of the kidney is that it regulates hormone secretion. So the kidneys would receive an input that there is a drop in oxygen saturation in the blood, known as hypoxemia. And then the kidneys would integrate that, and based on that, they would secrete the hormone EPO, erythropoietin. And then EPO would increase the red blood cell formation, and that would increase the oxygen carrying capacity in the blood. So it is a hormonal regulator. It is also important for fluid balance. It will modify accordingly the reabsorption and the secretion. So we know that in the body we have how many compartments? We have two compartments that are actually three and two. We have an intracellular cellular compartment, and we have an extracellular compartment that is divided into two compartments: the vasculature and the interstitial fluid compartment. So we have the interstitial fluid compartment and we have the vasculature. Who has the most amount of water? Who has the most amount of water? It is the interstitial compartment. Has about, how much does the interstitial compartment have? It has about 65% of the water that's in the body. The remaining 35%, 25% of that is inside the blood vessels, and the rest is found in your interstitial compartment. The interstitial compartment, what is it? Is it water? No, it is a thick gel layer, because it has a lot of proteoglycans, and that is very important. What is the role of the proteoglycans? It is a defense mechanism. It's a safety factor against the formation of edema. So when we say a patient has edema, it means the patient has accumulation of water. Where? Where does edema happen? A lot of water in the vasculature, a lot of water in the interstitial fluid compartment, or a lot of water intercellularly. If my patient has edema, that means they have a lot of water accumulating where? Accumulating inside the cell, intercellularly, or accumulating in the interstitial fluid compartment between the cell and the vasculature, or accumulating inside the blood vessels. Inside the blood vessels? Good, but not true. Yes. Yes, in the interstitial compartment, very good. This interstitial compartment is actually water, but it has a lot of proteoglycans that make it look like gel. So it's a gel, it's a thick layer. It is watery, but it's thick because it has a lot of proteoglycans. And that is a safety factor. Why is that? So if water leaks out from the intercellular compartment, it pushes it back inward. And if water leaks out from the vasculature, it pushes it back outward. That's why if you have edema, you must really understand that there is a really big problem leading to, maybe there's so much osmosis, there's so much solutes causing a severe oncotic pressure, leading the water to flow out, or maybe there is a defect or a hemorrhage or a congestion of the blood here that is leaking out through the interendothelial gaps and causing edema. So if edema happens, that means you really have defied this safety gel proteoglycan layer. So your kidneys are very important for regulating the fluid balance, regulating the intracellular and interstitial and intravascular water that's found within your body. So the kidneys, by the reabsorption from the lumen into the basolateral side, we regulate the water flow. So we manage hypovolemia and we repair hypervolemia and we restore euvolemia using the kidneys. Then the kidney is also important for electrolyte balance. So we said that we exploit the sodium potassium ATPase at the basolateral side to create a gradient for the passage of sodium from the tubule of the lumen. From the tubule of the lumen where there's urine, you have a lot of sodium influxing, and we use that to power the rotors, to power all of the channels to bring in the sodium, and with it glucose, with it also the other solutes and amino acids. And we also use it for acid-base balance, because we use that sodium gradient in order to send out into the tubule lumen for acid-base balance, to send out the hydrogen protons. So the kidney has multiple functions, multiple functions, more than just two. You know, there's a hormonal function, and it's like a gland, it's fluid balance, electrolyte balance, acid-base balance. But don't just memorize them, you must be able to connect how they function. What are the components of the renal corpuscle? That's really important. So the renal corpuscle is a 2-in-1. The renal corpuscle, it is made up of a vascular compartment. So the renal corpuscle is made up of a vascular compartment, meaning blood vessels, and the vascular compartment means my glomerular capillaries. It is made up of tubular compartment, which is my Bowman capsule. So the Bowman capsule is a tubule that is connected with the proximal convoluted tubule and the descending loop of Henle and the ascending loop of Henle and the distal convoluted tubule and then the collecting duct. These are all tubules. These are all tubules. And then on the other side you have all capillaries. So now the interface where they both combine is your renal corpuscle. You have the vasculature and the tubules. And the tubules are trying to make urine, fluid 1, while the vasculature is trying to make fluid 2, which is blood that is going to ultimately go to the peritubular capillaries and then back into the right heart. Starting with the renal corpuscle, list the components of the renal tubules as they are encountered by the filtrate. Well, that's easy. So I have my renal corpuscle, and then I have my Bowman capsule. After the Bowman capsule, you have the proximal convoluted tubule. Then you have the descending loop of Henle. Then you have the ascending loop of Henle. Then you have the distal convoluted tubule. Then you have the collecting duct. Okay? Another thing, we have the descending loop of Henle. It's thick at the top, thin in the bottom, and then thick again at the top. So what does that signify? If you are thick at the top, really thin in the bottom, You see? If you are thick at the top, thin in the bottom, what does that signify? What does that signify? Round 2. What does that signify? If in your descending and ascending loop of Henleys, a portion is thick and the other portion is thin, what does that signify? Are we clear? Yes. You mean to decrease the flow for exchange? That's very good. Intuitive answer. That reminds us of what happens in the lungs. Remember? So in the lungs, we have very good intuition. So what happens in the lungs? We have a constriction that happens here in order to slow down the passage of blood. And when we slow down the passage of blood, we maximize the exchange of carbon dioxide and the uptake of oxygen. So this is what you meant. Good guess, but wrong answer. So here, what are we trying to maximize? You mean, so these are the tubules, and we have the peritubular capillaries. So you mean that we slow things down so we can maximize the uptake here? No, because what's happening here is that we don't even have these protein channels here. That's why they are so thin. They are so thick because that portion has all of these big channels. The sodium potassium ATPase, the uniporter that takes sodium and glucose with it. Takes sodium and amino acids with it. And here we excrete. We take sodium in and we excrete out hydrogen proton. So these channels are thick. That's what makes them thick. This thin part is water. It's just for water. So the thin part signifies that this is the region that it doesn't need a lot of channels. So it's just for water reabsorption. But the thick region, because it has a lot of mitochondria, and it has a lot of activity of these protein channels on both sides, on the basolateral side and on the luminal side. You really need to understand the luminal side, the basolateral side. Who is at the luminal side? Who is at the basolateral side? How do we power the movement of all these solutes? That's very important. You guys understand? You guys understand which channel is mainly at the basolateral side? What is the channel that is at the basolateral side? What is the channel that's at the basolateral side? Yes. What channel? What channel? You mean the capillaries? Channel. A channel. The channel that's here at the basolateral side. Okay, I think I gotta say it one more time. The capillaries are channels? You guys think the capillaries are channels? Okay. I have in red my blood vessels, my tubular capillaries. On red, my blood vessels, my tubular capillaries. Then, what else do I have? I have the luminal side, where the urine is forming. This is the luminal side, where urine is forming. Urine. This is the luminal. This is the luminal, also known as the apical side. And these are epithelial cells. These are epithelial cells. These epithelial cells are connected by gap junctions. So now this in the core is urine, and these are my tubules, and here is my capillaries. So this side is the basolateral because there's a basement membrane here. That's the basolateral. That side is the basolateral, and these are my blood vessels. So what happens is, for my reabsorption, I have just here finished my filtration. I need to take glucose in. I need to take glucose inward. I need to reabsorb glucose. I need to reabsorb amino acids on both sides. I need to absorb glucose from the lumen. I need to reabsorb amino acids. I need to absorb my solutes. I need to reabsorb my solutes. How am I going to do that? I'm going to utilize sodium movement. Sodium movement. So I'm going to utilize sodium movement to bring in glucose. I'm going to utilize sodium movement to bring in amino acids. I'm going to utilize sodium movement to bring in the solutes. I'm going to utilize here sodium movement to bring in the solutes. Utilize sodium movement to bring in amino acid. Utilize sodium movement to bring in glucose. But what is making sodium move in this direction to begin with, so it can help me? Remember, I mentioned to you in the waterfall. In the waterfall, what's happening is we're at the waterfall. And water is falling. MGH. So you put a rotor here and you put the blades out, the water is going to hit the blades, causing the rotor to spin, and you put a magnet inside and a solenoid and whatever, and that will make energy for you. But here we exploited an already moving current of water. Here we already have a moving current of sodium. What created that movement of sodium in these directions so I can couple with them and shuttle with them all of these other solutes? What is making sodium go in this direction, in this direction? And then I'm using it as the rotor. I'm using it as to spin my rotor to bring glucose in. I'm using sodium moving there to spin. I know glucose is moving in because it's hitchhiking a ride with sodium. Amino acid, hitchhiking a ride with sodium. Solutes, chlorine and phosphate, hitchhiking a ride with sodium. But what is making sodium move from the lumen to the apical side? From the lumen apical side into the basolateral side? Yes?
Is it the concentration gradient?
Yes, it's the concentration gradient. But what is making the concentration gradient from this direction to this? Why not that direction? Why not sodium is moving from here to here? Actually, it should be. It should be moving from here to here because sodium is a lot in the blood vessel. Sodium is a lot extracellularly, in the extracellular compartment. In the interstitial compartment, there's a lot of sodium. It should have been the other way around. It should have been, glucose should have been dumped out in the urine. Amino acids should have been dumped out in the urine. So sodium should have been flowing into the lumen, not out of the lumen. What is making it going out of the lumen? Excellent. It is the sodium potassium ATPase channel that's here. hydrolyzing ATP. It's hydrolyzing ATP to take all the sodium that's coming inward. She will take it, throw it more into the blood vessels where there is a lot of it already. So it disturbs the gradient. So sodium comes in, she will say, no, go out. So now there is less sodium. So these guys will say, there is not enough sodium, let's flow in. And the sodium flows in, she will say, let's go out here. And she will send him out. And then the sodium potassium ATPase will use active energy of ATP to deplete the core of the epithelial cells of the tubules and make it minus 70 millivolts, make the electric voltage for sodium so low to ensure there is always sodium flowing in, so I can couple with its movement in my reabsorption of my amino acids. Not only does it do that, it also, for excretion of hydrogen proton, it uses sodium going inward to push hydrogen proton outward. Is that point clear? It uses sodium flowing inward for counter transport. So this is unitransport and this is counter transport. But in the end, I use secondary active transportation that has been facilitated with primary active transport sodium potassium ATPase. Yes, Eric?
So it's also the sodium pump that's located on the bottom.
So yeah, so it's the sodium potassium ATPase at the basolateral side. She is the one who's getting paid ATP in order to disturb for me the sodium gradient and ensure there is always an electrochemical gradient. There is a diffusion gradient. Sodium needs to move from a higher concentration gradient area to an area of a lower concentration gradient. And also there is a chemical gradient to move from an area of a higher electronegativity to an area of a lower electronegativity. The sodium potassium ATPase at the basolateral side ensures two things for me: electrochemical gradient, so that I can power the movement of sodium from the lumen into the basolateral side, and power with that a rotor protein channels that will reabsorb sodium, reabsorb solutes, reabsorb amino acids, reabsorb the glucose and other solutes, and also secrete out for acid-base balance, secrete out into the urine the hydrogen proton. So only examine if I ask you, what is the only channel that, although it uses secondary active transport and uses and exploits the movement of sodium down concentration gradient, but it is a counter transporter? It does not take the substance inward, it takes it outward. You should say that is the sodium hydrogen proton counter transporter. It takes the sodium movement inwardly to power the movement of hydrogen proton outwardly into the urine. Is that clear? Describe the effect of decreasing the afferent arteriole radius on the glomerular capillary pressure and filtration rate. So I know that there is a relationship. I know that there is a relationship between the glomerular filtration rate and my afferent arteriole, and I know that there is also a direct relationship between GFR and urine production, urine volume production, and glomerular filtration pressure. So they are all directly related. You increase urine volume, that means you have increased glomerular filtration rate, that means you have increased glomerular filtration pressure, that means the afferent arteriole has increased. But for the efferent arteriole, I have an inverse relationship. So the relationship will be that the glomerular filtration rate, glomerular filtration pressure, urine volume production will always increase in a relationship that is directly proportional to the inverse of the efferent arteriole. On the exam, I'll ask you, write these two equations. So depicting the relationship between the glomerular filtration rate and the efferent arteriole. So for the efferent arteriole, you do this. Glomerular filtration rate is directly proportional to the efferent arteriole radius, while the glomerular filtration rate is directly proportional to the inverse of the efferent arteriole. I have spelled it out, and I am afraid some people are still not gonna put it right. Describe the effect of increasing the efferent arteriole radius. We outlined which one is a direct relationship, which one is an inverse. So direct means when we increase, everything increases. When we decrease, everything decreases. And for the efferent arteriole, it's an inverse relationship. If we increase one, everything decreases. If we decrease the efferent, then everything else increases. Okay, describe the effect of increasing the efferent radius. We talked about that. Continue. Okay. All right. Generate the PDF and please send it to me by tonight. If you have not purchased the Physio EX and you are not on my list, just the Physio EX, not on the Elaine Marieb.